Regulation of nucleocytoplasmic mRNA transport. The regulation of nucleocytoplasmic mRNA transport refers to the mechanisms that control the movement of mRNA molecules between the nucleus and the cytoplasm in eukaryotic cells. This process is crucial for the proper localization and expression of mRNA molecules and involves several steps and regulatory factors. Here are the key aspects of nucleocytoplasmic mRNA transport regulation. First is transcription-coupled export. The process of mRNA export from the nucleus is closely linked to transcription. As mRNA is synthesized in the nucleus, it undergoes processing and packaging into a ribonucleoprotein RNP complex known as messenger ribonucleoprotein mRNP particles. These mRNPs contain not only the mRNA molecule, but also proteins involved in mRNA processing, transport and translation. Transcription and RNA processing factors facilitate the assembly of mRNPs and their subsequent export. Second one is nuclear export factors. The key regulator of mRNA export is the nuclear export factor called the heterodimeric export receptor complex consisting of NXF1 nuclear RNA export factor 1 and NXT1 nuclear transport factor 2, like export factor 1. These export factors recognize and bind to specific elements within the mRNA molecule, such as the mRNA5 cap and the poly A tail. They facilitate the translocation of mRNPs through the nuclear pore complexes NPCs, large protein channels that span the nuclear envelope. Third is RNA binding proteins. RNA binding proteins RBPs play crucial roles in mRNA export regulation. They associate with mRNPs and can interact with export factors and other components of the export machinery to influence the efficiency and specificity of mRNA export. RBPs can recognize specific sequence motifs within the mRNA molecule, known as mRNA export elements, and promote or inhibit mRNA export by modulating the interactions between the mRNP and export factors. Number 4 is post-transcriptional regulation. Several post-transcriptional processes can affect mRNA export. For example, alternative splicing can generate mRNA isoforms with different export properties, allowing for the selective transport of specific variants. Additionally, RNA modifications such as methylation or pseudoredylation can impact mRNA export by affecting the interactions between RBPs and the mRNA molecule. Number 5 is signaling pathways and cellular state. mRNA export can be regulated in response to various cellular signals and environmental cues. Signaling pathways, such as the stress response or developmental signaling, can modulate the expression and activity of export factors and RBPs, thereby influencing mRNA export. Changes in cellular conditions, such as changes in temperature, pH, or nutrient availability, can also impact mRNA export as part of the cellular adaptation responses. Sixth is quality control mechanisms. Cells have quality control mechanisms to ensure that only properly processed and functional mRNAs are exported to the cytoplasm. These mechanisms include surveillance and degradation of defective mRNAs, such as those containing splicing errors or premature termination codons, to prevent the production of abnormal or non-functional proteins. The regulation of nucleocytoplasmic mRNA transport is essential for proper gene expression, cellular function, and development. It allows cells to control the localization and availability of specific mRNAs, ensuring their delivery to the appropriate cellular compartments for translation and function while maintaining the integrity and fidelity of the mRNA export process. Thank you for watching, and don't forget to subscribe. Like and share.